And what I'd like to do is introduce you to cephalopods. And I love this. I used to remember watching otters come up with Coke cans and bite them in the Monterey Harbor and suck out the octopuses that were lurking in there. And I don't want to embarrass Steve, but well, I'd like to dedicate this simply because he's been such a friend, such a great educator and interpreter and visionary. Baja is one of the places that the Monterey Bay Aquarium actually took cerebral form. He's a wonderful diver, photographer, facilitator. If he were in Japan, he would be Ningen Kokyu, which means a prized treasure of cultural traditions. Anyway.
Oh dear.
Way back in our early education, we knew there were plants and animals. Later we found out, oh yes, bacteria. And then, and even Darwin was one of the first to draw a tree of life. And I love, this is from one of his journals, and he starts it by saying, I think. Ernst Haeckel drew one of the classic trees of life. Well, from a simple tree, we now have this massive, almost indecipherable domain listing of animals, or of life, not animals. You wonder where animals are, they're there. So this is mostly what we understand based on genetics and everything else. And when we look at animals, there's still the classic classes and phyla, and we're going to be talking about mollusks, which sounds strange. They are the second most abundant invertebrate animal. Of course, the most abundant are arthropods, and I think that's probably going to hold true until we start really looking at the ocean. Once we really look at the ocean, we'll probably find more and more things. Mollusks are soft-bodied. You're very familiar with most of them. They usually have shells. There can be 2 shells, a bunch of shells. Sometimes they've lost their shells. And we're going to look at basically one class. And some of these have lost their shells. These are the cephalopods. And mollusks are ancient, they're diverse, 100,000 living species, and that's why we're going to spend so much time talking about all of these. Everyone's familiar with terrestrial ones, but you may not be familiar with all of the marine ones. They do have a head, and a head is a really nice thing to have. And without a head, you don't get a head, because that gave you a bilateral symmetry. It allowed you to move, allowed you to concentrate senses, and we can thank a little primitive worm for that. And mollusks have a head. They're bilateral. They don't have segments. They have a muscular foot and a secreting layer called the mantle. They do have a blood and mostly an open circulatory system, except the cephalopods, and I'll talk about that. And radula, they have a mouth that basically has a rasping file inside of it that's mobile, and that's why they can eat so much, and they lick with their tongue. They can be herbivores or carnivores. Most of the ones that we're familiar with are hermaphrodites. That is, they have both genders. This is the radula, and it's worth it sometime to take a look at a snail. Hopefully it's not eating your strawberries. Most of this is made out of chitin, which is the same material that, say, crabs are made out of, but sometimes there's iron embedded with this. So it is very, very hard. And this tongue actually moves and can lick, and while it licks, it can drill holes. So some are incredible that they can lick completely through a clam. And cephalopods have this still. They do have eyes and a very complex nervous system. And from the beginning, the nervous system was basically in the interior and separated. That is, there were a couple of different brains. It depends how you count them. Cephalopods may have up to 9 brains or more. We'll talk about that in a minute. Most mollusks have a specialized free-swimming ciliated larvae, which is also shared by the worms, which says there is an evolutionary relationship between the two. This trochophore larvae often turns into a planktonic free-swimming form called the veliger. And these are very important in the ocean because they become a critical part of what everything else eats. They float around, they can swim a little bit, but they're nice, tasty little morsels. And cephalopods aren't the only things that have conquered the ocean. And I have to nod to these wonderful little creatures. They're basically free-swimming snails. Some of them have remnant shells. Some of them carry no shell. You can see this. Oh, I have to find out where my pointer is. But the one in the top, actually, you can see it. It still has its remnant shell. These are variously called sea butterflies, heteropods, or pteropods. The little red one here is called the sea angel, and they dance with each other when they're mating. So they represent a very old attempt to swim in the oceans. Here's a sea butterfly as well. Now, we're going to be talking about a lot of time. And I'd love to do this. You've heard about the time, a clock being used to represent life on this planet. I like this one. If you put your arms completely out, then about where your wrist is where the first animals begin to appear. So one arm, the one fingertip is when the earth began. Over here on your other arm, the wrist is where animals begin in the ocean. At a particular time, they become really abundant. And if you want to grab a fingernail file and file off your fingernails or clip them, then humans disappear. The early ocean was still filled with life, multiple cellular life, metazoans, but we wouldn't recognize anything. This is what is called the Edicarian fauna, and we don't even know what a lot of these things are. And then very suddenly, new forms appear, very big, strange forms. I love this little creature. When people first saw the fossils, they said, "What is this?" And they named it hallucinogenia, because you have to hallucinate to order to understand it. All of these things are wonderful. We think we know what these are. It's a type of worm. But something happened about 500 million years ago, which is about where your wrist is. And suddenly, it's called the explosion. I like to call it the Cambrian celebration, because suddenly, very quickly, all sorts of animals that we would be familiar with appear, including the mollusks. It's everything that we know today appeared about 500 million years ago. It's still a mystery. What caused this? We know very early there was a complexity, and of course this happened in the oceans, and secondarily later occurred on land. Here's another just graphic illustration of how quickly life suddenly diversified. And one of the theories is that algae and plants became abundant, or at least algae and oxygen increased in the oceans. And of course, we begin to see the first cephalopods. And the very first cephalopods were actually these little sort of flattened, almost, if you know what a limpet is, a single shelled thing that crawled. And then they began to get like dunce caps, and these dunce caps may have closed off into chambers, and then this chamber was light and allowed them to float. And so they lifted off the seafloor, and then that chamber got longer. And those were the things that lasted for about 200,000 or 200 million years. And most of those are fossilized. So the ones we know today are very recent. They're really from about the end of, or the beginning of the Cretaceous period. This gives you some highlights of what developed, the coiling in terms of cephalopods, and then later these chambers, and also a very specific nervous system. One of the causes we really only can surmise, but oxygen concentration, and using oxygen, animals could then break down sugars, carbohydrates, get more energy. Terrestrial erosion probably fertilized the oceans. There were changes in ocean levels and so on. We really don't know what, except it has been legitimately put forth that some sort of seeding took place. We had life forms, but not of this complexity. And people have found both proteins and also semi-nucleic acids in comets and asteroids. So we may have been seeded. In fact, there's a lot of people think that the aliens that brought this seeding were the original cephalopods. Certainly when we look at alien movies about aliens, we like to portray them as what? As cephalopods. And we'll talk about that a little bit more. Here's an idea of what might have happened. Little lift off, beginning to coil. Maybe some of them became crawling on the bottom. We don't really know. And maybe later, these things fell out of their shells and became octopi or octopuses. And I should mention that octopods are a made-up word in the sense of combining Greek with Latin. So to use the proper plural would be octopuses. So I will probably make a mistake a lot. It doesn't matter. One of the first squid-like animals had this very strange set of sort of chambers in its mantle, and these were hollow. We know them from fossils. These things got big, they got massive. They had tentacles. The tentacles were often spined. And you can see the one fossil in the middle that has a fossilized chamber, and this is called the belemnite, a belemnite. And these things got big. Imagine seeing something like this 30 feet long. There've been 30-foot ones found. And these probably were an adaptation to regulate buoyancy. They look super heavy, but the shell itself was very thin, aragonite probably. And then these were air-filled or fluid-filled chambers. And so they could adjust by pumping water or air in and out, and they could move throughout the water column. Many of these became incredibly abundant and became very, very important in the fossil record. These are known as Orthocyrus. They're mostly from Morocco, but they're found in various forms. They're beautiful. They often get carved into jewelry, but originally these were hollow inside of a squid-like creature. that got enormous. Sometimes they opalize really, really nicely. And you can see a little hole in one of these, and that was where the air or the fluid moved in and out. Lots of people like to collect these because, and throughout history, these were thought to be very sacred sort of things. When you found something like this, what did it come from? Nobody had any idea about squid in those days. And once you got really long, who knows, the genes began to express themselves and twisted these long bodies and shells into all kinds of shapes. These are called baculites. When the animal twists and turns, and you can see in some cases they get very invaginated, and that kind of strengthens the curving area. And then they just let loose. They curl back on themselves, they twist around. So you can, it's hard to imagine a squid-like animal living inside this thing, but it's not as heavy as it looks because it was a thin shell and then a hollow set of chambers inside. But these became ridiculously ornate. We know mostly from the fossil record, and these are beautiful things. Steve's been to my house. He knows the house is littered with these, and I hope sometimes I can share them with you. We're working on a way of doing a show and tell. These are just remarkable. They look so heavy, they should, but they were hollow with the chambers, very light, and they maybe danced across the floor. And this may have been an adaptation too, to prevent themselves from being eaten. This is how the fossils are often found in various places. They just fell to the bottom of the ocean for whatever reason, got melded into the muck and mire, and then fossilized. And so you can see how many were fossilized at once. Unbelievable diversity, some of them armored. Believe it or not, one theory about the shells that like Diplomacyrus, it may have been like a paper clip so that they could clip themselves onto something and not have to swim all the time. And of course, you know, this is a sort of a sequence. So you see a straight one, and then you see it begin to coil and coil again, and then eventually they coil around into something we would recognize as a group that became very, very prevalent, nautiloids, and then later another group called Ammonites. There are shows, and I should tell you, and I'll tell you again later, every early February in Tucson, Arizona, there's a gem and mineral show, a fantastic celebration of past life, and people from all over the world bring their fossils and hold reconstructed dinosaurs and gems and so on. So you can buy almost anything you want there. These are what are called ammonites, and I'll tell you the ammonites are a little different than nautiloids. Nautiloids are very old, but ammonites are subtly different in their diversity and also how they filled and emptied their chambers. But they coiled, they twisted, they did everything. And they eventually became quite fast swimming. And one of the differences is that the little siphuncle that allowed the air to go in and out is sort of at the base of the coiling. I can't find my arrow, otherwise I would do this. And you can see that it has something that we would call a gill. Gills, there's a stomach, a mouth, a funnel. The funnel acts as an anus. So here's the anus. This is the beak, and inside the beak would be a radula. We'll talk about that again. But this is how you tell an ammonite, where this hollow tube, which empties the chambers inside. As a fossil, these get filled up with quartz and calcites and all sorts of beautiful things. And They are just gorgeous. They're made into, people cut them and make them into all sorts of jewelry. Now, this is an old rule. Edward Drinker Cope was one of the great men of early science from Philadelphia. He was independently wealthy, and he began to publish about fossils. First publication when he was 19, he ended up publishing something like 14,000 or 1,400 publications in his lifetime, digging dinosaurs out in the West, fighting with his competitor named Marsh. But Cope noticed that the more recent fossils would get incredibly large. And this is a group essentially related to scorpions called Euryptrids. Earliest forms in evolution, very small, they get bigger and bigger. And horses, the earliest horse was the size of a terrier. And then they got bigger and bigger and bigger. Dinosaurs started out very small, then they got bigger and bigger. Triceratops, its baby was, again, it's not its baby, but the first Triceratops was something equivalent to a small dog. Here are trilobites, very ornate. This is a millipede. How'd you like to meet that in a dark night? This is a dragonfly that would probably buzz you. And this was a scorpion, what's called a water scorpion. Cope's rule applies even to Godzilla and to King Kong. The earliest movies back in the 50s, he was pretty tame. And you can see up here, Godzilla and King Kong are now bigger than they've ever been. So gigantism happens even in the movies. But to give you a sense of the gigantism, there's a diver, this is a scorpion, and look at those little pinchers. And here are what are called belemnites, and they float. And ammonites, they got rather large. And here's Camosirus that got very, very large too. And this is in the late Cretaceous. This is fairly recent when ammonites became very abundant. And people still, if you actually, what's the name of the movie? It's Kate Winslet, I think, who plays, oh, I can't think of her name, who lived in Lyme Regis in South England. And she would collect shells and polish them and occasionally sell them to the museum, the British Museum. And there's a nice little movie about her and Lyme Regis, and people are still discovering all sorts of things there. So you imagine this sort of cephalopod swimming around the ocean. It would be hollow, but it would be a pretty tasty meal if you could get by the crunch. The little thing, tongue twister, she sells seashells by the seashore. It was all about Annie. The kids would make fun, and as they went into her shops to buy whatever they could. This is called ammonite. It's a recognized gem. It's one of the few gems that animals have made, although the animal didn't really make it. The animal made the multi-layered shell that got filled with opalized material, and then the layers refract this. And ammonite, it only comes from southern Alberta in the Bear Paw Formation along a river. They're mostly encased in this shale and mud, and they're fun to dig up because once you take that stuff off, this just lights out. This little piece would probably set you back about $60,000. I have a few small ones I hope to share with you. And you'll see these kinds of gems made every once in a while. So ammonites enrich your life, even in their depth. And of course, not only did they get big, they had to get armored, and for very good reasons, because in the late Cenozoic, there were dinosaurs in the ocean everywhere. Mosasaurs, ichthyosaurs, well, not so many plesiosaurs, but all of these fed on cephalopods, which were the most abundant things. And quite often you will come across cephalopod fossils with rows of teeth bites in them. these are the teeth, and look how big some of these cephalopods, or these mosasaurs, got. They too diversified, they too got gigantic before they went completely extinct. This little one here is an ichthyosaur, which you notice is basically converged on a dolphin, except for the tail. They became alligator-like, just massive things. They could eat anything they want, and they ate lots and lots of ammonites and nautilus. So here is a sequence of what probably happened from this little flat thing that became a single tube. They began to curl. Maybe that was harder to eat, and then they got bigger and more massive. So here you can see the chambers inside, and then eventually the coil. This is actually a nautiloid because the siphuncle that drains the water or the air is actually in the middle. So nautiloids, which are actually much older, and somehow they survived. We don't really know how they survived the great extinctions, and there were multiple extinctions. They have this siphuncle in the middle. You see all the chambers are hollow. They do have gills, they do have a beak, and they have what they're called tentacles, and they're unique because they have up to 100 or so tentacles, which is pretty impressive. These make beautiful fossils in the sense that they can be opalized on the outside, filled in, and so on. One thing that happened, it's probable that the nautiloids began to live very, very deep in the ocean, and that may allow them to survive the massive extinctions that occurred. In the ammonites, one of the things we see is this crazy suturing in the shell. This is just a part of the suturing, and that greatly increased the strength so that they too probably went to the deep, deep ocean, and this allowed them not to be crushed, but something eventually did them in anyway. Why the nautiloids survived and the ammonites didn't, we will never really know. But the nautiloids, way, way back 400 million years ago, 400 million years ago, are still with us. The ammonites and ammon refers to the spiraling horns of ammon, and belemnites may have survived by becoming modern, the modern forms of cephalopods. And the history of life is also the history of extinction. And here is a Cambrian explosion, but there was an event in the Ordovician, there was a big event in the Devonian, and then a massive event in the Permian. Some of these due to huge outbursts of lava flows, like in India, that actually increased CO2 and may have acidified the oceans and dissolved all of these shells. And then we come up to this, to the Triassic Jurassic event, and then this is what we're sort of going through now, or exacerbating it. What went extinct? The marine ammonites, all the marine reptiles, more than half of the corals went extinct. All the dinosaurs went extinct, pterosaurs, a lot of the flowering plants. Anything over 25 kilograms probably went extinct. Trilobites disappeared completely. Only the nautiloids survived. This is a great story that is not over, unfortunately. There's a gripping book called The Sixth Extinction, which is on now. So if we look at the Cambrian explosion, we see very simple primitive forms coiling more. There's the nautilus that makes it. Then they coil and become hollow, chambered, floating things. And many of these go extinct, and we're left with just a very few. Now, Why did all this happen? Well, one thing, there are major genes that are sometimes erroneously called master genes. They're called Hox genes because they form a particular pattern called homeotic boxes. So that was shortened to Hox genes. These are crucial. They develop limbs, they develop segmentation, they develop all sorts of neat, groovy things that make the animal what it is. And early in the mollusk evolution, there was a freeing of these Hox genes so that one set of Hox genes could work on the dorsal surface and another one worked on the ventral surface, but these became elaborate. This allowed for heads to develop, eyes to develop, gills, and 3 hearts, and so on. So there are genetic causes, but what caused this in the first place? Maybe the Cambrian environment changed sufficiently so that these genes multiplied, or they segregated somehow in the larvae. Cephalopods. Most everyone's familiar, and I don't want to go into all the details because the taxonomy is still really difficult to work with. And so for our purposes, we're just going to say nautilus, octopuses, and squid. How's that? And I'll tell you the reasons for those. And vampire squid, because they don't quite belong with anything else. They're all marine. There's never been a terrestrial form. They have a head surrounded by a foot, which is modified into arms or tentacles. They're mostly 8 or 10 in number. Tentacles are found primarily in the squid and the cuttlefish, and they have suction cups and very highly modified teeth or grabbers or hooks to catch your prey. They produce ink. They have beaks made out of chitin. They have a closed circulatory system as opposed to an open system, which makes them metabolically much more capable. And they have separate sexes, and they tend to be short-lived, which is an enigma in itself. They may only live 5 months, rarely some live 5 years, but they do have complex nervous systems with one of the best eyes ever produced in evolution. So we're just going to talk about the nautiluses, the cuttlefish and squid, and we'll throw the vampire squid in there, and then octopus. Those are all the terms we really need to know. Squid and cuttlefish have tentacles, and these are very long and extrusible. They can shoot out. They're hooked, as you can see. Some have very elaborate, different kinds of suckers, often with the tooth inside, a chitinous tooth. Some are gluey, the hooks and so on. And these can be shot out. Here's a little cuttlefish shooting one out. Here's a larger squid that's going and grabbing these. And this pattern is even found in the colossal squid. So tentacles are distinct from arms. The tentacles can be shot out. It's like if you hold a water balloon and you squeeze it, one of those long ones, it will extend. And that has to do with the musculature. You have circular muscles and longitudinal muscles, and the arm is filled with muscle and fluid. So it is very much like a water balloon, and it can be quite rapid. So octopuses have arms, and they're very distinctive in a pattern. The squid have tentacles and arms, and they also will then have 10 arms and legs, or 10 arms and tentacles. So they're called the decapods, and only octopus have 8. So they're called the octopuses. And here, this cuttlefish has both arms and tentacles, and then the nautilus, which has these 90 to 100 different arms or so. even the very biggest squid may have these incredible hooks. I mean, if you're going to be grabbing something large, you want to hang on. And so these hooks, here are teeth that are chitinous and ring the suckers. So you can see these are also flexible and mobile suckers, and the suckers on the tentacles are incredibly movable, and they can compress and they can form grips, not only hooking grips, but sucking grips. And one of the ways they do this is that their muscles, the closest analogy, believe it or not, is our human tongue. That is, we can protrude our tongue, and that happens because there's muscles at the base, there's muscles around the edges, and there's muscles through the middle. And if you get the circular muscles squeezing the internal muscles, your tongue will extend. And then there's nerves that will control all of this. And then underneath, the same nerves will control part of the sucker. And there's a circular ring, another circular ring, and a ring like this that will actually, whoop, sorry, I have to be careful what I touch. So if you watch what happens with the sucker, and if the sucker disc is ever attached to you, there are circular muscles here that can open up and attach flatwise. Then they kind of create a vacuum in here. Then they can push this down and then expand it this way, so you get a really tight hold and sometimes very difficult to detach. And these are often on little peduncles so that they can be moved in different directions. Now, there are other kinds of arms, even in octopuses, and these are called Hectocotylus arms. Steve will talk more about these and how they're used. Originally, they were thought to be parasites, but they're very much involved because you would find them singly attached like this on something like an Argonaut or another squid. But it turns out that those are used in transferring sperm or sperm packets, and I'll let Steve talk about that. So here are some bobtail squid that show modified arms for transferring sperm or sperm packets. The brain, there's a central brain, and interestingly, the esophagus runs right through the middle of it. So the esophagus coming up like this goes through the digestive glands and then comes out the funnel. And there's two huge optic lobes. So if you want to count them, there's 1, 2, 3. And then there's a circular ring, and each tentacle has a basic long axon that is independently capable. And not only are these independently capable, so here is a nice diagram. The main brain is not a donut, but it's just to show you what the esophagus is like going through this, and then the optic lobes. And there's also a little structure just underneath it called the statocyst, which I'll tell you about in a minute. In the original optic lobes here, which are quite large, there may be 60 million neurons in each one. But don't get too excited, because once you include the peripheral arms, there's anything from 250 to 320 million neurons. This puts them on the level of sort of a dog in terms of the neuron numbers, but also perhaps their teachability. These peripheral ones can contract, they can bend, they can grasp, and I did have it written here, I don't know what happened. They also have sensory nerve endings, so that the suckers can be used not only to attach, to hold, to pull things in, but also to taste. And also probably to remember, because all along these arms are little mini ganglia. going to each of the sucker discs. So they remember, say, someone that they've touched. There are many stories Steve can tell you that some octopuses will touch their feeder or touch somebody and not like them. And every time that person comes back, they squirt water at them. You know, they don't like your taste or whatever it is. There's also two stellate ganglia back here that control the internal operations. So it gets complicated. These are independent brains, but you can see all the little nerve endings that are going out to the eye. Inside the brain, or just adjacent to the brain, is one of these remarkable structures that people aren't aware about. It's a hollow area, and inside is a little calcified stone that's suspended. And jellyfish develop these things. And depending on how the jellyfish moves, the stone will actually move and trip these suspensions and trip nerve endings. And it tells the jellyfish where it is in time and space. If you're living in a three-dimensional world, you can go up, you can go down, you can go to the side. Most of us can only go that way or that way. We have a limitation. But if you're in a three-dimensional world, you can go up, you can go down, you can go anywhere. So these statocysts are pretty well developed. We also use them because we can age them. We can use them to age the animal. Strangely, we still have these inside of us. Inside what is called the cochlea, there are little ciliated things, and sitting on top of it, let's see, where's a good stone I can put up there. And when you shake your head like this, that stone is going to move and lean on the nerve endings and tell you where your head is. So proprioceptors, especially in the head. The eye, which we think is incredibly complex, and it is, although people have figured out it could happen in 300 generations, maybe something like 200,000 years, and an eye could develop. It's not all that complicated, but what is unique, and there's a lot of things that had eyes. One of the first things was a little flatworm, and then octopuses have a camera eye, and the reason for that is if you look at the octopus eye, right, there's a lens, there's an iris, there's a cornea. Not all of them have everything. Nautiluses don't have the cornea, and they have very squinky little eyes. It's almost a pinhole camera, but the lens is there, very protein rich. What's different is this would be the film. Right. In vertebrate eyes, the film is in the back of the eye, if you see over here. And what's out in front are the nerve endings, which is sort of backwards. And it also produces, since the nerves come in the back of the eye and spread, it also produces a spot where the pigment cells, the rods and the cones, don't occur. And so in vertebrate eyes, there's a blind spot. And Steve will probably talk a little bit more about that. Why did the eye develop so well in cephalopods? There is a major gene called the Pax gene, and the Pax gene is very, very old. Insects use a different form of gene, but squid have has the Pax6 gene, which is used to develop the optic cup and a bunch of other things. But they have multiple variants. They have over 4 different variants of the Pax6. So this probably caused an elaboration of the eye. Even mammals do not have the same variation. So again, genetic reasons behind a lot of these changes. There is a buccal cavity in both cephalopods and both squid and octopuses, and inside both of them is a chitinous beak. And chitin, again, is what insects are made out of. It's also found in mushrooms. It's a universal protein. And this is used for nipping and biting. And inside is the radula. So here is the buccal cavity. They're calling it pharynx. It's the buccal mass. The beak's inside, and then the radulus is there. And these beaks can get pretty big, and they can give you a pretty good nip. In octopuses, it's inside the buccal cavity. There are the beaks, and they pivot and rock and can just nip. And they can also stick out their radula and lick holes in shells. And then there's a digestive gland. The radula also chops things up so that they can eat things a little better. The ink gland. Maybe you were capable of getting a squid to dissect in class. You cut this here, and there was a little protein-rich rod, a pen shell or gladius, which is found in squid. And you found the digestive tract, and then you'd see this silver-lined thing that had ink inside of it. And the gills would be out here. Well, these are Jurassic ink sacs. This little painting was done by scraping this open, getting the ink, revitalizing the ink with water, and drawing. It was a very popular thing to go down to Lyme Regis and dig these things out. By the way, the ink of cuttlefish, the scientific name for the cuttlefish is sepia. And the ink is really beautiful brown. So the name for that color, brown, is sepia from the animal's name. All these will ink, and there's multiple kinds of inking. Sometimes it's just to confuse a predator, sometimes just to make a morph. People have tried to study it, but it's a little diverse. Ink is made out of a lot of different things. Melanin is the dark substance, which is a tyrosine-derived pigment. There's sugars, there's proteins. Look at all the amino acids. Boy, you could drink this, and believe it or not, squid ink is a medicine in many parts of the world. There's fats, and there's also cadmium, lead, and copper, which is sort of interesting that there would be metals. They get this primarily from their food and also from bacteria. Here's the pathway that produces melanin, and it's based on tyrosine, and also produces DOPA, which is a neurotransmitter. Sometimes it's quite surprising, and sometimes the amount of ink coming out, and they're not the only mollusk that inks. This is a little giant sea slug called a sea hare, and they occur here, and they're very abundant, say, in Elkhorn Slough. And if you agitate them, they just release this beautiful purple ink. I used to dye a t-shirt in it, and it would hold fast. Now, there's another creature that eats squid almost exclusively. And because it eats so much squid and so much ink, it also inks. These are the dwarf or the pygmy whales. And I love to show you this skull, which is just absurd, as all dolphin skulls are. And this is a pygmy whale, a pygmy sperm whale, because there's no teeth on the upper maxillary, and all the teeth are on the lower, just like a giant sperm whale. And this is ideal for holding on to slippery buggers. This is hooked and so on. And this is the sea hare. This is the color of the pygmy squid or pygmy sperm whale. And this is the color of the cuttlefish. I've been caught in this a couple times. We call it poo-nados. Sperm whales eat really big squid, and they must probably use this for communicating, or just sort of a whale raspberry or something. If they don't like what the others are doing or saying, they'll let you know. And it becomes quite colorful. And you can see it sometimes floating. We used to collect it to try to analyze it. And also later we learned that we could get the skin from these whales because it sloughs off, and then we could do DNA fingerprints from these whales rather than analyzing their poop. I will talk a little bit more about cephalopods as food, and because there's a tremendous amount of animals that eat and depend on cephalopods, but in the bottlenose in the eastern or the western Pacific, they will often capture cuttlefish, jam them into the bottom, and then kind of take them up and then cause them to release the ink, and then come back and rub it until they get the cuttle bone out of it and the skin off of it. But sometimes dealing with octopuses can be rather tricky. So one of the treatments that dolphins will often do is just slap the heck out of them, throw them around. There is a record recently, I have a picture of it somewhere, of an octopus, a giant red, actually suffocating a dolphin that didn't quite know what it was dealing with. It covered its blowhole and wouldn't let it open its mouth. The old kind of chestnut, what has 3 hearts, 9 brains, and 8 legs. Well, that would be a cephalopod, 8 or 10 legs. There is a heart called the systemic heart that pumps over to the gills, and that pumps through the gills and back and out. So this is kind of deoxygenated heart. They use hemocyanin, which is a copper-based, if you see this copper there, and it can be deoxygenated. When it's deoxygenated, when it's coming out, it's clear. When it's oxygenated internally, it's blue, because oxidized copper, if some of you may know, blue or copper sulfate, which is used in pools and so on. There's some evidence that in deep water animals, this actually reduces, it serves as a antifreeze. And it is, the only time I've ever seen it is if you're cutting a lot of squid, your knife blade or something might be blue. But there's a lot of interest in this in terms of possible medical uses. There are lots of gill arrangements. Long ago, the squid generally just has 2 long, big gills, and this is where the oxygen exchange takes place. There's 2 big arteries or vessels that will pick up and drop off. Now, none of us live alone. We're learning more about our intestinal bacteria. Well, squid and octopuses have all kinds of bacteria, not only in their intestinal tract, but in some cases they can be put in different spaces. And one of the things that they get from these bacteria is a neurotoxic poison called tetrodotoxin. And tetrodotoxin has a certain shape that can fit into the side of a nerve and block the channel where sodium would normally go through to act a nerve response. Well, some of this stuff is stored in the muscle or organs of certain fish, and also certain salamanders like this, which is hopefully come out tonight in our first rain. Come on, rain. Or blue-ringed octopuses. And of course, this is well known from fugu. Japanese chefs are so adept at cutting the liver and making beautiful arrangements, and then you could eat the fugu and get a pretty good buzz. It will affect the heart, the nerves, the sensory nerves, all sorts of things. A few thousand people die every year in Japan from fugu poisoning. That's all right. You got a good buzz. And actually, the FDA allowed it to be brought into the United States, but it's anyway. Or you can just lick a pufferfish. There is a very strong association between fugu from pufferfish being dried and blown into people to mimic death, blown into their nostrils. So go from fugu, from pufferfish to fugu to zombies. How's that? There's, of course, some things that are very, very toxic. All cephalopods basically are poisonous. However, only a few of them will kill you if they bite you with their beaks and lick you with their tongues. So, and they usually advertise this fact. Very common and very polite of them to say, look, I'm very gaudy. That means I'm probably very toxic. So the blue ring octopus, it's still using tetrodotoxin, the same thing that poison arrow frogs use primarily. And there is a difference in that, and venomous are toxic. Here's a blue pinstripe pajama squid, which is a bottle-tailed squid. And it's one of the few, I can't even think, maybe Steve can, that has a permanent pattern. And the pattern is actually advertising the fact that if you eat this, you're going to die. This doesn't have to bite you. This is completely toxic. And again, it's tetrodotoxin. Likewise, the flamboyant cuttlefish, and this is a cuttlefish, which is so cute, and Steve will talk about them because they're just lovely creatures. They too, all parts of the flesh are poisonous, and probably they advertise this fact. Now, I'd sent out, and Michelle was so gracious to send this on. There are some very good books, public or popular books. I listed them by popular and academic. Peter Godfrey Smith is very interested in intelligence and also mind, whatever that is. These two books are good introduction, and this is a meditation on consciousness. And I always loved this because it was one of the first books I ever read on this, and the subtitle, The Soft Intelligence. And he spent a lot of time, and he had a great deal of respect. He was one of the first octopus learners instead of an octopus teacher. And also a local, Dana Staff, who did her graduate work at Hopkins Marine Station with Bill Gilley. She's a lovely speaker. She basically writes and does talks. And Paleo Nerds is a group that actually has interest in the past. And she has written, actually, these are the same books, essentially, but this is one of the best introductions, and she talks a lot about evolution. There are a number of other books, as I said, and these two books I just include because they're great books on mollusks and seashells, and a couple of them have a little chapter on argonauts or something else, but if you like shells and so on. And Cynthia Barnett is also wrote a beautiful, beautiful book on the rain. And she lives in Florida, in Jacksonville, not Jackson Hole. Anyway, and lots of academics. If you want to support become friends of the Marine, Hopkins Marine Station, you sometimes get library privileges, and there's some great books there. So, boy, we've run out of time, and I'm only halfway through. I haven't really introduced you to all the great creatures. So maybe that's why we stuck another lecture at the end, so we can maybe elaborate on sentience and intelligence. Steve will tell us more about the life histories, but I hope then I can tell you a little bit more about how we interact and what we think of octopuses and squid, and also what they mean to us and what they do for us. There's a lot of medical advances that are coming out of different things. The beak of cephalopods has a unique structure that's being used. The cuttle bone of the cuttlefish has been used to show us how to package things better, light and strong, and so on. Tetrodotoxin is now being applied to use as a painkiller, and there's on and on. And just the whole field of neurology would not be possible without squid. Not to mention people love to eat them and are eaten and probably overfished, and for various reasons, not just for food, but for ornament.